Well, welcome everybody to the joint RSS PSI webinar on the topic of Estimands. I'd like to start off by thanking our sponsors, Quintiles and Wiley. Both of these are long-term supporters of initiatives to share statistical thinking more widely, and we certainly appreciate their support yet again today. We've got two great speakers. We've got Dr. Alan Phillips, I should say an industry veteran in my experience. He's VP of Biostatistics at ICON, and he'll be talking about current developments in Estimands, particularly the PSI expert group on Estimands and sensitivity analysis and a paper that was published in Pharmaceutical Statistics. We then have Professor Peter Diggle from Lancaster University, who is also the current president of the RSS. And Peter will be providing some observations based upon historical work from a red paper in 2007, but predominantly his current thinking on the topic. And we'll introduce a term called predictand, which is an interesting one, so a new one to me today. I'm now handing over to Alan Phillips. Thank you, Alan.
Okay, thank you, Andy. I'd like to thank everybody for attending and taking the opportunity to hear about the PSI EFPI Special Interest Group on Estimands. For those of you who may not be familiar with the pharmaceutical industry, PSI is an acronym for statisticians in the pharmaceutical industry. Hopefully the RSS are aware of that with the potential merger. They're a body of about 1,000 statisticians working in various phases in the industry. And EFSPI is the European Federation of Statisticians in the Pharmaceutical Industry. And it's really a body of national countries, so PSI, the French, the Dutch statisticians all go in through EFSPI. I'm just trying to make sure, oops, I've gone right to the end. I don't want to do that. So what I'm going to do in the next 30 minutes is to just give a background of how the special interest group came about and also what happened, to look at what is the problem we're trying to solve here as a group of statisticians and try and illustrate that through a case study, look at some of the key messages that came from the special interest group, and then make a few closing remarks. So for those of you who work in the pharmaceutical industry, back in 1998, which is some time ago, ICHE9 was issued, entitled Statistical Principles for Clinical Trials, and it's almost like the Bible relating to some of the issues that we face as statisticians in the pharmaceutical industry. It's been an excellent guideline in it, and that it's stood the test of time. But one area that has had significant debate over the years relates to missing data. So back in October 2014, an addendum to ICHE9 was proposed relating to estimands and sensitivity analyses. And as I say, it was based on it arose from the back of the discussions of missing data. For some of the analyses that are done, missing at random is often assumed for patients who drop out. Now, the missing at random assumptions assume the missing value mechanism can depend on the observed data, including any responses, but given these measurements, there's no remainder dependency on unobserved responses. So, in its simplest way to look at this can be in a clinical trial or a longitudinal study, that two patients who share the same treatment and covariates and exactly the same response measurements up to the point at which one of them may drop out and the other remains in the study. The missing data from the subjects who dropped out have the same statistical behaviors as observations from the subjects who remain. And there are a number of people who say, well, actually that assumption can't be valid because by the definition, patients who drop out are different. So based on the back of discussions over several years, this ICH addendum was proposed. And when we get new regulatory guidelines, professional bodies such as PSI, EFSPI offer to help. So we formed an industry group to try and come to a consensus on the subject. We sent out for volunteers in August 2014, some time ago. And our first meeting was back in February 2015. And I've listed the people down below the 13 people who attended the first meeting, and it's their views that we're discussing today. So it's not just myself. So the credit needs to go to all these people. Although we had a meeting, subsequent discussions carried on in the virtual world through emails, and eventually we came out with a position paper, and I forget the date on that. But it's these people from the various pharmaceutical companies whose work we're presenting today. The work was fed back to the ICHE9 Addendum Steering Group through two representatives we had on there, Chrissy Fletcher and Frank Brett. And for those who are interested in any guidelines, if you want to go to the actual addendum, if you go onto something like the EMA website and you search for various things like ICHE9, you will pick up the addendum, and it looks like this. So when a new guideline comes out, what the professional bodies such as PSI try and do is set up a special interest group, working group, whatever you want to call it, to try and debate the issue to come out with an industry consensus that we can then feed into those guidelines as they're being developed, so that it's not too restrictive for industry but provides good scientific statistical principles. For this one, we really looked at three streams of discussion. What we would normally do is set up some questions for the members to debate and try and come up with consensuses, or where there's differences of opinion, to record those differences together with the reasons why around certain topics. And for Estimands, we decided that we'd do three areas. One was going right back to the beginning, such as what is an estimand, what is it, and why should we even be bothered with those. If you go to the concept paper, they do provide some examples of estimands, two that they actually give in the concept paper: difference in mean outcome improvement for all randomized patients, and difference in outcome improvement in those adhered to treatment. So they give some examples of what they mean by estimands. But we asked this expert group or special interest group, is it as simple as just rolling these out for every clinical study that's conducted? That is, can a generic set of estimands be developed, or will every study require something that's different? If you can develop a generic set, what would they be? From either an efficacy or safety? Would it vary by therapeutic area? Does oncology have unique needs? So we asked those fundamental questions in the context of estimands of what is it that we're trying to do here, and how far do we need to push it. On the anticipation that we would come out to a common consensus on what they were, we then looked around how would you go about implementing them in the framework of clinical research. So what level of details would be required where. For those of you who are not familiar with clinical research, for every clinical study that we do, we write a protocol. And that protocol is fairly detailed of why we're doing the study, what measurements are being collected, when they're being collected, how the data will be analyzed. The principles of the statistical analyses are detailed in the protocol. We then create something called a statistical analysis plan, which has more details around it. So we debated things like the implementation. Should you actually be doing the details of estimands in the analysis plan versus the protocol? How many can you have per study? When we come to look for marketing drugs, we combine studies. So really, should the estimands be at the combined level, the integrated level, looking at the totality of evidence, or should it be at the study level? So we asked a series of questions, like on the screen, around the implementation aspects of estimands. Finally, the last area we looked at was around education and communication. Clearly, if estimands are going to take off, we need to look at the advantages and disadvantages. The medical community can tend to be very conservative about adopting new methodology. So if we were going to get this out and it's an advantage, we're going to have to publicize that. Are statisticians and non-statisticians on board with it? So what sort of education are we going to have to do around estimands, both within the statistical community and also the non-statistical community? And then you've got this word estimands, which I tend to think of as a statistical, a mathematical type word. how will that go down to physicians, pharmacokineticists, etc.? And how do you explain that then in the context of these non-statisticians involved in the drug development process? So there were three main areas that we focused on as a group. I'm on, for those people who are remote, I'm now on slide 8. So we started by looking at a definition of what is an estimand, trying to get everybody onto the same page. And quite simply, if you go to something like Wiktionary, an estimand is very simply defined as that which is being estimated. So although it tends to be a statistical term, I would say more an estimand more akin to statisticians than a physician, the concept behind it is fairly straightforward, that which is being estimated. If you go to reports that are available for clinical research, such as the National Academy of Science report in 2010, it talks about the trial protocol, which, and this definition is taken from that, which should explicitly define the objectives of the trial, the associated primary outpoint or outcomes, how, when, and on whom the outcomes will be measured, the measures of intervention effect. So, you know, when you go to these, you kind of think, well, why do we really need estimands? It should be defined, the protocol should be fairly explicit. So where do estimands actually come into play? For those who have worked in the pharmaceutical industry and clinical research for some time and have seen a broad spectrum of protocols, frequently you tend to find that the study objectives, even though contrary to what's being requested, lack clarity on details. It's not uncommon for somebody like myself or somebody who gets to see lots of protocols to see a statement as the objective to compare the efficacy and safety of drug A versus drug B. They're very, very high level without any specifics. The trouble with that is it often results in a misalignment between the planned study design and the statistical methods. And then what is required to be estimated for the primary objective questions of interest can actually then be misinterpreted by the statistician or misaligned. The other issue that we tend to see in protocols is the assumptions being made in both the design and the analysis are not clear. So what assumptions can be made for patients who drop out? How do we want to handle them? What assumptions can be made that are relevant to the analysis? They're often not clearly defined in the protocol. The other thing that we do, which ICHE9 is built around, is robustness of inferences. I'm sure you'll all be pleased that drugs don't get registered just on the basis of one finding. We tend to look at robustness of those inferences to different types of analysis. We tend to call them sensitivity analyses. And without the assumptions being clearly, explicitly documented in the protocol or even in the analysis plan, the sensitivity analyses supporting the primary inferences from a study can also be misaligned. And quite often, it's not uncommon to find that sensitivity analyses, actually when they're conducted, make the same assumptions as the main analysis. So although people think they're doing sensitivity analyses, all they're doing is repeating the analysis in a different format. So the first conclusion that the group came to is there is a need for estimand. There's a need to improve the clarity between the objective statements written such as to compare the efficacy and safety and what is planned to be estimated as per the definition of estimand. So that was good news to get the team off to a good start. When you look at the literature, there is a definition that is available on estimand that the group liked. And really the way to think of the estimand is a more detailed objective statement. It's going to be very difficult to change some of the practices within the pharmaceutical industry to change some of the objective statements, although that would be ideal. So the way that we were viewing this was to have a more detailed objective statement of the estimand, which reflects what is to be estimated via the scientific question of interest. And the definition that we came upon was that by Bretz and Akea. And they really had three components to their estimand. First component was the population of interest. second component was the endpoint of interest, and the third component was the measure of intervention effect. And the working group liked this definition and felt it's one that could be moved forward by statisticians involved in clinical research. A couple of points on the definition. It should be said that the population of interest shouldn't be confused with the study or analysis population. The population of interest in the definition is that for which we're assessing the scientific question of interest in the concept of drug regulation. It's basically the population that is intended for post-approval use. So it's a slightly different concept. Measure of intervention effect. I'm not sure I personally like the term of measure intervention effect, but it was the best one that was around. And by this, we mean that the issues that need to be taken into account for potential confounding due to post-randomization events, such as, it's probably best illustrated by examples. You can get potential confounding of the treatment effect due to non-compliance. Some patients may or may not take the medication, and it might be different in the different groups. Discontinuation of study medication, the dropout patients, treatment switching, rescue medication. These are the types of issues that need to be thought up front of how they're likely to impact the estimate of the treatment effect, either positively or negatively. So the definition that we honed in on was this one that we liked. Andy said I'm an industry veteran. I like to think of myself as quite young still. But there were quite a lot of us all of the same sort of area in that meeting. And we thought, well, we'll have a go at trying to define an estimand. So we started out with a case study. We challenged the team to come out with a case study of these people. And the one that's often selected to illustrate some of these issues is a trial in diabetes. And so the case study we chose was a randomized two-arm trial, drug A versus drug B in diabetes with type 2 diabetes mellitus. For these type of studies, the endpoint is change of HbA1c levels, and often it's collected between baseline and 24 weeks. So assessments are taken at 4, 8, 12, 16, 24 weeks. For ethical reasons, we assume that patients could be switched to rescue medication. So once their HbA1c values were above a certain threshold, they could be switched to rescue medication. But apart from that, for this case study, we assume that all patients would be followed up for the whole duration of the study. And that in itself is the first interesting point, that we assume that there were no missing observations. So although the concept of estimands arose from the debate around missing data, this implies that estimands is actually independent of missing data, how the missing data are handled. That estimating the estimand in the presence of missing data becomes a technical rather than interpretation issue. But anyway, this was the case study that we started on. And the 13 of us thought, oh, we've all been, we've seen these types of protocols before. So we kind of understood the study objectives. It was the comparison of drug A and drug B in diabetes patients. So we then started turning our attention to trying to define the estimand. And we didn't get very far before we first got into trouble. So we started by saying, well, who is this study really aimed at? So is the study being designed to make statements targeted at patients? Is the study designed to actually look at payers or regulators or sponsors? Because depending on who you are or what the study is, your estimand might be different. So we quickly, having gone through quite a lot of these before, came out with two potential estimands for this study. The first one was we were aiming it at more along the patient to say, well, if I start off in this study and I take drug A plus rescue, how does that compare for me as a patient versus drug B plus rescue? And this tends to be known as an effectiveness estimand. However, from a sponsor perspective, they probably want to know what's the effect of drug A whilst on treatment versus drug B whilst on treatment. So they're probably not interested from including the rescue medication in here. So very, very quickly, after coming to a conclusion, we knew what the objectives was, we then have to stop and think, well, actually, who is the trial aimed at? So we're going right back to square one here, and we honed in on these two estimands as potential ones. So we overcome that hurdle after a little bit of discussion, and then we started thinking, well, actually, you know, let's talk about endpoints. What is it that actually we're trying to do here? Are we trying to look at the second estimand, drug A whilst on treatment versus drug B whilst on treatment? So in other words, we're looking at the response at the end of treatment, or are we looking at some kind of response or average response throughout the study? So through these discussions, trying to define and come up with a statement of estimands, it kept on drawing us back to the objectives and kept on trying to think about what we were actually trying to do. We often take too many things for granted. You know, we've been in here, there's a lot of pressure, there's been numerous diabetes trials set up. We kind of know how to do them, but do we really know how to do them? We then started talking about the rescue medication. How does that come into it? How do dropouts come into it? And through this very, very simple case study, we started peeling back the layers of the onion here. Just every time that we took one step forward, somebody came, why? And it actually then opened up to what are we really trying to do here? What type of assumptions are we going to make here? Because until we fully understand what we're trying to get to, we can't home in on the assumptions. So through this case study, we came out with some very quick learnings from here that actually for a single clinical trial, there are many possible estimands for a set of objectives depending on where you sit. And really, the statement, the estimand statement, that to which is to be estimated will be dependent on how you're going to use the results. So you're going to use the results to target patients, to target payers, regulators, sponsors. Also, as well, from the estimand's perspective, it kept on going back. It wasn't here's the objectives, now write an estimand statement. It was a case of, it was an iterative process that we came out. We came out with, you start with the objectives, then start thinking about the comparison of interest. Then you look at the study population, then potentially the endpoints, and you're continually reiterating around a loop to try and refine things here. So it's got to be a dialogue. It's got to be a multifaceted approach to defining estimands. But we felt through doing this exercise that there were benefits because it would help everybody in that clinical study team to understand what they're really trying to do and then how that links to the analysis. It also helped to hone in on what data you have to collect post-randomization. So trying to hone in on the estimand and write it down. Do you really need to collect data post-discontinuation or once a patient has dropped out? Or is it just adequate to let them go? And again, it goes back to what is it that you're trying to estimate here? Are you looking at an effectiveness type of analysis where it's drug A plus rescue versus drug B plus rescue, so the treatment regimes, or is it the efficacy type analysis, just looking at drug A without the confounding stuff? So we proposed an iterative framework for defining estimands, starting with the trial objectives, writing the estimands. Potentially you have to go back to more information on the trial objectives. What are you trying to do? An iterative type of approach. Then you start looking at the study design, what's being collected when. That may in itself start looking at the trial objectives. And then so it's an iterative sort of process all the way down to the analysis method and the sensitivity methods. For those of you who are interested, last year there was a publication looking at the choice of estimands and analysis in diabetes trials. This came out post our little exercise. But it gives some insight into potential estimands for the the case studies considered. And the variable in time point here is HbA1c as measured on the average glucose control over the preceding 5 to 12 weeks assessed after 24 weeks. So it wasn't at an end point, it was controlling over a period of time. And some of the measurement of intervention effects that we started discussing, that they outline here quite nicely. So here they've decided that the actual treatment comparison is that of the assigned treatment as randomization. So where rescue medication was taken, they wanted the hypothetical values if rescue medication had not been taken. At the bottom, for losses to follow up, hypothetical values if the patient had continued to take part in the trial. In the case of discontinuations, the actual off-treatment values. So if you want more background, I go have a look at this paper. This actually gives more detail and is quite a useful read. The interesting thing here, patients are followed up even after the study treatment for these types of estimands, and there's been a lot of discussion in the clinical arena about whether or not you should follow up patients. And also the paper looks at different potential approaches to the analysis, which I won't go into, but different types of approaches, completer analysis, carrying forward the last pre-rescue analysis methods, considering missing at random as an assumption, etc. So it's a useful reference if you want a little bit more background. I mentioned that we asked the first question of who's the trial aimed at. So I would just want to expand on this. Depending on where you sit in the clinical trial merry-go-round, regulators are probably looking at questions like how a treatment works whilst the patient takes it. They probably want to look at the evaluation of different policies. Payers will probably want to look at questions, what would have been the treatment effect if the patient hadn't switched from the randomized treatment, or how the effect of the treatment varies depending on the order in which it is taken. Patients, being a patient, I've taken medication, I want to know what's happened to me if I start this treatment. What will the treatment benefit risk be if I take all the treatments as directed? Sponsors show patients who will probably look at things like how does the treatment work in clinical practice, extrapolating from clinical trial environment. So each one of these people, which has this different type of question here, may potentially have a different estimand that needs to be worked out with different assumptions, different types of data being collected. I mentioned earlier about the efficacy, estimands, and effectiveness. Effectiveness is basically looking at the treatment regime and is sometimes known in the literature as de facto estimands. If you look at efficacy, it's sometimes known as de jure estimands. There are some publications out there that actually say all you really need to do is to look at the de facto estimand, but the special interest group felt that actually that should not constitute the sole basis for a regulatory decision. There are these other perspectives that need to be built in and is important when assessing data from clinical trials. Just moving on, I'm aware of time. The ICH addendum concept paper also talks about sensitivity analyses. I mentioned already that ICHE9 tends to look at robustness of inferences. So how robust is that? But the expert group liked this concept of sensitivity analysis by Lusch, who basically defines sensitivity analysis two different types, internal validity and external validity. So internal validity for an estimand is where the model assumptions are changed, but you're still looking at the same estimand. External validity is where actually you're changing the estimand and looking at the robustness of whether or not by changing the estimand you still get the same sort of inferences. Now, I guess some people, is actually changing the estimand really a sensitivity analysis, or is it a different type of analysis? But generally, the concept of looking at these two approaches, internal versus external, we felt was the right way to go, changing model assumptions, maybe changing the estimand slightly to make sure that the inferences that are drawn from a clinical trial for which marketing authorization is granted is the right way to do it. The final one that I said we'd look at was education and communication. we concluded that actually, in order to get this moving, a cultural change for the pharmaceutical industry in those in medical research would be needed, and a new way of thinking would be required. It was interesting, even at the meeting, the statisticians didn't actually totally come to consensus. So even within the statistical community, there was a need to come to a consensus before we sent it out. But if we were going to be successful in rolling out Estimands, it's going to need a pull from the regulators. So when they actually receive studies which have objectives like to compare the efficacy and safety, for them to push back and say, well, what do you mean here? You're not helping us. You've got to define it a little bit more. And also, we as statisticians in the pharmaceutical industry need to go out and start promoting best practices and standards and start showing them examples of what we actually do. There was concern about whether ICHE9 statistical principles was the right place for this, because to define an estimand, you really need to have all stakeholders involved in a clinical trial involved in that process. By linking it to ICHE9 statistical principles for clinical trials, by actually having, the word estimands, whether it would turn our clinical colleagues cold, and whether somewhere like ICHE8, which is general considerations for clinical trials, might broaden it. So there was discussions on that. However, it was felt that there was a clear need that we had to change. That the study objectives would benefit, and the clinical trials would benefit from actually better linkage between the objectives and the analysis, and the framework would enable to do it. It also leads to more thought going into upfront the collection of data, what needed to be collected when. So even just looking at things like patients who discontinued, by better defining what is being done in the assumptions, you can then decide whether or not you need to collect data post-discontinuation or not. So in the interest of time, some of the key points. So the problem statements that Estimands, in order for the addendum to be successful, the feeling was that the problem statement that Estimands are trying to solve needs to be clearly formulated. And actually they're there to address deficiencies in the study design and objectives and to link to the primary analysis, so there is a need there. I guess ideally you could improve the objectives, but we were realistic and we didn't think that would happen, so there is this need for Estimands. There is a working definition that can be used by everybody, but Estimands will be a multidisciplinary problem. It's not just the statisticians. It's not to be passed over to just those quirky statisticians. So you've got to have an understanding and engagement from all those involved in clinical research, from design, conduct, and analysis. And one of the things that isn't available yet, it's beginning to now with that paper, is good shared examples. We don't believe there will be a standard set of Estimands. Each study is slightly different. There may be some therapeutic specific Estimands. So, for example, oncology, you might come out with some. But really, ICHE9 addendum needs to focus on a framework for developing the Estimands by all those involved. And because clinical trials are multifaceted and expensive, that by definition there is going to be more than one Estimand per study, which of course, from a statistician's perspective, you've got multiplicity issues that need to be taken into account, etc. But generally, whenever there's going to be a label claim, so a claim that goes on the label for a new drug, there should be an upfront Estimands defined. Sensitivity analysis already defined, there's a good definition. We refine that to comparing different estimates of the same parameter versus comparing estimates of different parameters. We felt that both were needed to check the robustness. There couldn't be any pre-specification of how many. We need to have sufficient changes, sufficient variations in the model such that there's confidence in the conclusions being drawn from the study. Finally, in the interest of time, don't underestimate the cultural change that's going to be needed here. As I said, there is no shared understanding among statisticians. So the first step is to promote a common understanding among the statistical community, for those people who are involved in study design, drug development, etc. And then once we've got that common understanding with good case studies that we can then go out and share them with our clinical colleagues in multidisciplinary teams to build them up. But it's going to take a lot of effort and a pull from the regulators and a push from us people within industry to move the statisticians, to move clinical research to better defining the analysis methods, the assumptions that are needed, etc. I've tended to focus this presentation on clinical research, but I think the same principles applies to other areas of research. It's always good to make sure you understand what you're doing, why you're doing it, what assumptions you're making, etc. So I think some of the principles here also apply to other fields of research. With that, can I thank you? I think I pass on to Andy or Peter.
Yeah, over to me. Thank you, Alan. It's very interesting. So we're now going to pass over to Peter Diggle. And Peter is going to give his views on what he's heard, also with some historical context with his 2007 paper. So over to you, Peter.
Okay, thanks, Andy. I'm going to really make some fairly brief remarks, which are trying to sort of stand back from what I recognise are the very real and essential and crucially important wider issues of all aspects of designing, conducting, interpreting, and assessing policy decisions for clinical trials, but just to try to, in a sense, pick up some of Alan's themes, but put them in a rather specific, what I would say, a model-based context, which I hope will clarify some of the points Alan was making. It may give a slightly different perspective on some of them, but let's see where we get to. So if we could take the next slide, I'm simply going to show you the results of a rather old randomized trial of drug treatments for schizophrenia, and use these to sort of ask myself what's the question that these data might be trying to address, provide you with a model-based answer. The indefinite article is important there. I'm very much of the view that the way you build a model can and really should be conditioned by the question that you're trying to answer. I'll also make a distinction which I find useful in my own work between whether we're really trying to estimate something or predict something, and I'll give you a precise statement of the difference between those two things. And I'll then talk a little bit about something that I've already discussed informally with Andy before we started, about how what you think you're estimating can be materially changed depending on what you condition on before you try to estimate that thing. So if we go on to the first substantive slide, PANS acronym refers to Positive and Negative Symptom Scale, and this is a questionnaire instrument used by clinical psychologists and psychiatrists to assess a mental health patient's general condition. And this in particular is a summary of results from a drug trial of chronic schizophrenia patients who are randomized to one of three drug regimes. The black line relates to a placebo, the red line to a drug therapy that at the time of the trial was standard, and the blue line relates to a drug therapy that was being proposed at the time as a potentially superior treatment for this very serious and debilitating condition. Because this is a symptom score, low values are good, and the fact that in all three treatment regimes, the lines are going down, would seem to indicate superficially that the patients are benefiting from treatments. But note that that superficial statement applies to the placebo as well as to the two active drug therapies. So, if we move on to the next slide, what question might you want to ask of the data from that trial? You might want to ask what benefits a patient could expect to receive from either treatment. We might want to ask the question, what treatment should we recommend for general use? And there may be a number of other questions that we might want to ask. And Alan has gone into much more detail about whether these questions need to be qualified by statements such as, provided the patient adheres to the treatment, or on the whole, recognizing that some people do and do not adhere to treatment, what would be the overall benefit to the population of treated patients? And those are very different questions. So I'm now putting on this slide a full definition of a statistical model for the PANS data. And so far, I've only told you half the story, because the picture that I showed you a couple of slides back is a picture of the PANS measurements. The follow-up protocol for each patient is that they have their PANS scores measured at baseline, 0 weeks, and at 1, 2, 4, 6, and 8 weeks post-baseline. Okay? And you can see on the picture that there was a general tendency for the PANS scores to decrease over time. But what I was showing you precisely was the average score at each follow-up time in each of the 3 treatment groups for the patients who were still continuing in the trial. And what the picture did not show was that there was a progressive dropout throughout the follow-up period. Patients elected to leave the trial, and they were asked to give the reason for their leaving the trial, and the predominant reason was that they perceived an inadequate response to the treatment to which they had been randomized. And at that point, they were removed from the trial. The expression in curly braces is simply a quadratic in time to capture that apparent trend over the 8 weeks follow-up. And the alpha, beta, and gamma parameters are subscripted by the treatment to which the patient had been randomized. So, in effect, the term in curly braces is the formal model for the black, red, and blue curves that would be underlying the data in the picture that I showed you. When you analyze the data in detail, you find, as is typical of repeated measurement data of this kind, that data repeated measures on the same patient over time are not independent. They are serially correlated, meaning that observations close together in time are quite strongly correlated. Observations further apart in time are more weakly correlated. So the term S of T in that equation is recognizing that a person's PANS state, a person's mental state, will not follow precisely the quadratic curve, but it will fluctuate over time. And that induces a correlation in the data, because a patient will have spells in which they are feeling relatively better than the average for that treatment group, and at other times they will be feeling relatively not so well as others in that treatment. And those fluctuations over time are captured by the stochastic process S, which is that second term in the first equation. And then finally, we have a Z term because PANS is an imperfect measurement instrument. And so a patient's mental state is estimated by their PANS score, but it's not precisely their PANS score. So we add that Z term, which represents independent measurement error, or if you prefer, the inherent imprecision in the measurement instrument that's being used, and that gives you the model for the repeated measurements. And if we ignored the S term, we would have a simple quadratic regression equation. But we can't use that because the repeated measurement character of the data would make it an invalid statistical model. The second equation in that page is what I've called the dropout submodel. And that recognizes that at any time during the follow-up period, there is a probability, here Pij, that a patient will elect to leave the trial, predominantly, as I said earlier, because they see an inadequate response to their treatment. Now, I've written here a very simple model for that dropout process, and I'm not trying to advocate it as the best model for these data in particular, or for longitudinal trial data with dropouts in general. simply using it to make the point that when Andy referred to earlier to analysing data with a mixed effects model under missing at random assumptions, he was implicitly assuming a model of this kind, because that dropout model says in words that the probability that patient I will drop out at follow-up time J depends on what we have observed that patient, namely their PANSS measurement Y at the previous time, but not on anything that we have failed to observe. So in particular, it does not depend on their current mental state. Now that may or may not be a reasonable assumption, but it is the assumption that you are making if you adopt a mixed effects modelling approach and pass your data to SAS-Proc Mixed or to NLME if you're an R user, or to any other software that allows you to fit mixed effects models. We now get on to the topic of inference, or to put it another way, what question are we trying to answer in analysing these data? And again, I've picked an example, not because I want to argue that it's what one ought to be answering. It's simply one of a number of possible things one might want to know. And in words, it says, what's the difference between treatments 2 and 1 with respect to the change in symptom severity over the 8-week follow-up period? And I want to make the point that that is inherently a very imprecise statement. And all verbal statements, it seems to me, are inherently imprecise. And to make it precise, you have to refer that statement back to the model that you've chosen, your choice of model, and you have to then extract whatever features of that model are formalizing that question. Now, from an estimation point of view, my definition of an estimand is a very Spartan one. An estimand is a function of model parameters. In other words, Greek letters and known constants. So from that perspective, the average change between week 0 and week 8 in treatment 2 minus the same thing in treatment 1 is the function of the parameters that you see there on the slide, namely beta 2 minus beta 1 times 8 plus gamma 2 minus gamma 1 times 64, which is 8 squared. Now, a very different interpretation of the verbal question would be that the patient facing their treating physician across the doctor's desk might say, what will I experience as the difference in my symptoms after 8 weeks of this treatment compared to another treatment that I may have been on? Now, that involves not only the estimands that we previously discussed, so the betas, the gammas, the 8, and the 64, but it also depends on the change in their latent or random effect, which is the S process. Because on treatment 2, they would have hypothetically experienced an S2, 2 in brackets referring to treatment 2, and on treatment 1, they would have experienced an S1. And I haven't yet told you what assumptions I'm making about those processes, but the point is that they could be different. And indeed, they would be different in the real world because one patient cannot experience both treatment 1 and treatment 2 over the same period of time. So either this refers to 2 different 8-week periods, or one of these 2 terms in curly brackets here, the S2 8 minus S2 naught, or the S1 8 minus the S1 naught, has to be what causal inference people call a counterfactual. You can't observe both of them. Okay, so estimands are functions of model parameters, whereas predictands are functions of parameters and unobserved random variables. Okay, we're now looking at a logistic regression example in which I want to emphasize this distinction between estimation and prediction. And another way to think about this is to think about what you might be conditioning on when you want to analyze the effect of a treatment. Now, for visual purposes, rather than thinking of discrete treatments on this picture, I'm thinking of a continuous value of a covariate. So you could, for example, if you wish, think of this as the dose of a particular medication, and the logistic regression model is being used here because I'm thinking hypothetically of a binary response which you can imagine either you do or you do not experience a removal of the symptoms through a particular dose of a particular drug. And the black lines on the slide are indicating the responses, the probability of a positive response for a collection of patients who differ in the intercepts of their own logistic regression equation. Now that would either be because there's another patient-specific covariate for which you're adjusting, or it could be a random effect attached to each patient, so that some patients are inherently failing to respond, except at high doses. These would be the patients on the right-hand side of the sheet, black curves. Others are responding more effectively to lower doses for reasons unknown. The point about that is that if you are interested now in predicting the effect of dose changes for an individual patient, then because all those black lines are in their approximately linear path parallel, you can see that you are assuming that there will be an effect for each patient which is the same, and the slope of those black curves in their linear path is indicating how big that treatment effect is. If, however, you want to take a population-level approach to the problem and say, how would the average response change with a different recommended dose for a population, then you would get the red line. And it's clearly showing a shallower slope. And that's simply a byproduct of the fact that the model necessarily is nonlinear. And what you mean by the average effect of changing the dose is different according to whether you're dealing with the conditional response given the individual patient's random effect or covariate, and that's the slope of the black line, or whether you're talking about an effect averaged over a population, a heterogeneous population of people with different values of that extant or latent covariate, and that's the red line. But we're going to revisit the time data. And what I've now added to the previous slide is the fitted quadratics that were set out in the slide a few slides ago in equation form. And what you have are the fitted quadratic for the placebo in black, for the standard therapy at the time of the trial in red, and for the novel therapy at the time of the trial in blue. And one's first reaction to this picture is that it's rather ridiculous, because clearly the curves do not, in any ordinary sense of the word, fit the data. But rest assured, that is exactly what you would get if you process these data through SAS Mix or any other software, any other correct software for fitting linear mixed effects models. Because what you are implicitly estimating, so your implicit estimand in the model that's being fitted here under missing at random assumptions, is the mean response in a dropout-free population. And that may be a reasonable thing to do in this case, because, if you remember, the patients who dropped out were largely dropping out through a perceived inadequate response to therapy. And so it's not unreasonable to actually think, I wonder what would have happened if they had actually stuck with the program for 8 weeks. And the answer, to the best of our ability, is estimated by those quadratic curves. And those of you who are skeptical about the much vaunted placebo effect may be reassured to see that the black curve is indeed approximately horizontal. However, one can imagine situations in which the dropout was due to unacceptable side effects, in which case the value of these fitted curves is questionable, because it is hard to understand why anybody would want to know what the response would be in a dropout-free population if a proportion of those patients were in fact experiencing unacceptable side effects. So this is simply one illustration of how what perhaps at first seems a relatively subtle distinction between different estimands can actually have really profound, large effects on what conclusions one would draw. Now, that's more or less as much as I want to say, except to draw some concluding comments the data by making assumptions which may or may not be reasonable. So the art of statistical modeling involves a balance between the information you buy with your modeling assumptions and the risk you take in invalidating your inferences if those assumptions are grossly incorrect. And I say grossly incorrect because, as George Fox famously said, we know that they're not exactly correct and never will be. But my concern from my perspective of statistics is that when I see a procedure expressed purely in algorithmic terms, such as last observation carried forward, I don't know what it's estimating. I personally find it very hard to tie down a precise question unless I can formalize it using mathematics, by which I really mean mathematical notation. I do not mean mathematical theory. And I have to say that I have found in most of my work collaborating with non-statisticians that having a dialogue in which potential models are set out on a whiteboard, discarded, replaced by others, does actually, if nothing else, tease out the very precise formulation of the scientific question. My next point is that in doing this, I think it's helpful because it forces two things out into the open. One is to be precise about what you're trying to estimate or predict, and the other is what assumptions you're making in arriving at the estimation or prediction. And then the final point I want to make is that if you're dealing with nonlinearity, as you are in logistic regression, in survival analysis, in N+2, missing values, as you very often are in real trials or indeed in other scientific contexts, and serial correlation, which you almost always are with longitudinal data, then the combined effects of those phenomena are subtle and they can be counterintuitive. Okay, thank you.
So we're going to open it up now for questions, and also they can be done in two ways. They can be done via the phone, so by pressing *6 on your phone, or you can use the Q&A icon, and we'll get some questions coming through that way. So maybe I could just start off, though, sort of get us up and running. And I picked this point up from an SPI report. And it basically said MMRM, so mixed effect repeated measures, estimates the hypothetical effect if everyone would have adhered to treatment. And therefore, those that did not do well from a safety perspective. Can this method ever be a good estimator in an IT setting? And Alan, I don't know would you like to give your views on that? The web basically, can an MMRM estimate any good in a ITT setting?
So no model is perfect. Okay, that's what I would say. I would say that the MMRM clearly assumes missing at random, which, as I said earlier, patients who drop out are different than those patients who stay in the trial. So clearly, the statistical properties being inferred aren't necessarily going to be correct. However, how poor an assumption missing at random is, I don't know. So I'm not sure I'd ever go as far as to say it would never provide a good estimator. Intuitively, there will be deficiencies in the model, but how that performs relative to a better model, I don't actually know at this stage. But I wouldn't go as far as to say it can never be a good estimator.
Yeah, I mean, just one comment, risking a charge of pedantry, Alan. The fact that patients are different by definition because some drop out and some don't does not invalidate MAR. MAR is an assumption about the measurements you would have observed, or the properties rather, of the measurements that you would have observed. It's not in a certain sense, it's not saying the patients are equivalent. However, I do think it's, in practice, often at best an approximation to what will really happen. And primarily I say that because we make discrete time observations on continuous time phenomena. I think if we were able to monitor essentially in continuous time, MAR would be a very reasonable assumption to make in most cases. But because we are only monitoring in discrete time, in in my example of 1 or 2 week intervals, then in effect, anything that happened after the previous measurement cannot be captured by MAR. So I would agree that it's an assumption that should always be questioned. The serial correlation between longitudinal measurements acts as a partial rescue, because what happens at any given week is not independent of what happened previously. So if follow-up measurements were independent, then you would have no predicted value on what you would have observed currently had the patient not dropped out. Because they're serially correlated, you do have partial information, but I agree on the partial. On the specific question, it does seem to me that what I call mixed effects modelling under MAR assumptions, and I think you have a snappier name for that, which is an acronym I've forgotten. Clearly, it seems to me that is, as you stated, Alan, and as everybody as Andrew read out, it's making an inference about the population, a hypothetical population in which dropout does not occur. And equally, everyone is adhering to treatment. So it does seem to me it's fundamentally contradictory to ITT, which seems to me to have its origins in the primacy of the need for a valid test of the null, and in particular a randomization valid test of the null, as the treatments are equivalent in every respect. So I think ITT, almost by definition, must be addressing a different estimand than mixed effects modelling from the MAR assumption.
Okay. You mentioned the beloved LOCS, or the much maligned LOCS, which we all did many, many years ago. Again, I'd be interested in both your views on that. Do you ever see a comeback for that? I mean, Peter, it sounded like no way for you. You don't know what it is. And so, just interested to get your views on LOCS. Yeah, let's start with Peter this time.
Okay, yeah. My concern about LOCS, I think, is I would need a lot of convincing that it does anything other than provide one of many ways of securing a randomization valid test of the null. I'd be more sympathetic to last residual carried forward. For example, when you do have time trends, it seems to me that you might just about get away with saying we can extrapolate the time trend to those who've dropped out. And furthermore, because of this serial correlation phenomenon that's so prevalent, if somebody has been consistently above average throughout their period of observation, it seems to me to be plausible that they would continue to be above average in the future. But that's carrying forward the last residual rather than the last observation. And I think you can set the model-based version of that, which is in the Diggle, Farewell, and Henderson paper that is referred to in the introduction, is that if you assume that the residuals follow a random walk, then basically what you're doing is you're saying that in a random walk, the current value of the residual is the best point predictor of the future residual. So last residual carried forward, I think, has a defense. Last observation carried forward, I think, does not. But of course, they're equivalent if you're not seeing any secular time trends.
Okay. We've got a question coming through, but maybe I'll just get Alan, do you have any comments on LOCS?
So you say, will it make a comeback? From quite a lot of the work I see, I don't think it's totally gone away yet. I still see a large number of protocols with LOCF in, rightly or wrongly. That coming from some of the smaller companies, because I think it's appealing to the non-statisticians. It's simple in understanding. However, I believe we are moving into a direction where more complex modelling and missing data is the way forward. So I would like to see LOCF disappear to start with, and then you can worry about the comeback, because I don't think it's totally gone yet.
So Alan stated de jure estimands may provide important information to a patient and prescriber on efficacy if the treatment is taken as directed. But there are not manifold reasons why the product might not be taken as directed, and thus the patient might just as well want to know the de facto estimand without knowing that it's a bit more pessimistic. Do you have any views on that?
I think for a clinical trial, you're going to need more than one. I don't think it's just about efficacy. I don't think it's just about effectiveness. I don't think it's about just de jure or de facto. I think you've got to work out what you're trying to do, what the objectives are, and relate it. I think to have two black and white, which some of the literature boils down to, I think is not necessarily the right approach. So I think when you set them up, you do have to talk about the non-compliance. You do have to talk about dropouts. I know most of this was that the estimands came around the missing data, but I think the non-compliance is an issue. I think treatment switching is an issue which all needs to come into play.
Peter, any comments on that?
No, I except to agree with what Alan said.
Okay. I've got another question. This is directed to you, Peter. Trying to understand what Peter is saying, and he said he might be misinterpreting your point here. Does the concept of estimands and the testing of such go against the randomization principle?
It kind of does in the following sense that if you want to estimate something in my world, then that there has to be, even if it's not explicit, there has to be at least implicitly, and I prefer it to be explicit, a model within which that thing that you're trying to estimate is some function of the model parameters. Now, the good part about that is that you can then use the general theory of statistics to estimate that quantity with the maximum efficiency. Right? Rather than having an ad hoc procedure, you know that you can be fully efficient in your inferences. The downside of it is that you're making an assumption which you have to therefore be prepared to believe as a working approximation. And I really think it is an art, the art of the applied statistician, to make judgments as to when those additional modelling assumptions are worth incorporating into your analysis. When I was talking about randomization, I was alluding to the fact that, as I understand it, the reason why simple analyses are still popular in regulatory trials, and I understand this, when I say if I understand it, I agree with it, is that in order to have a guaranteed valid test of the null hypothesis of no treatment effect, then you need to be able to declare in advance an analysis protocol. And it follows that you cannot be model-based, because if you declare in advance an analysis protocol, and it turns out that that is a model-based protocol, and the data are demonstrably incompatible with that model, then it's clear that the analysis is worthless. What you can do is you can have an analysis protocol in which, at the very least, the test of the null is design valid. In other words, the randomization confers the validity of the test. So that seems to me to be a very conservative approach, which will be defensible in what you might call a quasi-legal setting of regulation. But the price you pay for that is that it does not tell you very much about what I think Alan called the efficacy of the treatment. In other words, it doesn't tell you what the treatment actually does, because it's not clear what your number at the end of the process, which is the observed difference between the two treatments. It's not clear to me, at least, what that's estimating. What it is doing is giving you a randomization valid test of the null. So it seems to me, as soon as you talk about estimands, you are at least implicitly talking about models. And if you're talking about models, you at least need to be prepared to check whether that model is at least approximately compatible with the data. It will never be perfect, as Alan's already said, but if it's grossly incompatible with the data, then clearly you can't trust the estimates that it produces.
Okay, thank you. Any questions on the phone?
Can I ask a question?
You can.
So, Peter, I was pleasantly surprised to say that when you talk with physicians, they like the model statement, the Greek letters and stuff like that. Is that universal?
They don't like the Greek letters, but I tell them what they mean, and what I force them to do is be precise about the question, because usually the first, I'm sure you must have this experience as well, usually the first approach says, I've got these data and I want to know if such and such a test is a reasonable way to analyze the data. And my response then is, tell me what question you're trying to answer.
Yeah.
And then they say, I want to know if there's a significant difference between these things. And I say, no, you don't. What question are you really trying to answer? And we then get into a dialogue. And I use pictures rather than Greek letters when I'm in dialogue. But I do find that it helps to create a shared understanding of what the real problem is, rather than them looking for a quick solution to a problem that they understand scientifically, but they don't understand statistically. and they don't therefore appreciate the pitfalls of simply going for a textbook solution, which may or may not actually address the question they're really asking of the data, or even if it does, they have no machinery for knowing whether the assumptions behind that textbook solution are actually approximately valid. And it has the ironic consequence for me that the people I'm talking about usually rush to things like Whitney, Wilcoxon tests because they're frightful of their data being non-normally distributed. When if they read Fisher, they would know that t-tests and f-tests are actually perfectly good approximations to the randomization distribution of a trial difference. And they're fretting about nothing. So it's really just a general way of trying to get away from a sort of a recipe book approach to finding the right method for a particular data set to analyze a particular data set, to a more question and answer based, saying what we're really trying to find out, what's an appropriate formal model within which we can address that question, and have the machinery also to check that the assumptions we're making are not grossly incompatible with the data.
And I think that applies to all areas of research, not just me, not just you, right across the board, I suspect.
Absolutely.
Yeah.
Okay, I have a question here from Sarah Bray, who was wondering if we need recommendations on how to report estimands in publications. Particularly, this would affect how we combine results across studies in terms of meta-analysis. So any comments maybe starting with you, Peter, on that?
Yes, I mean, I'm a rather skeptical of Bayesian formalism for scientific inference as opposed to decision making. But it's clear that when you produce an estimand, or to use a less fancy term, a point estimate, you need some measure of its imprecision. And what you are doing in a classical framework is typically, there may be some honourable exceptions, but typically you are implicitly assuming that the sampling distribution of all the parameters in your model is multivariate normal, and from that you can construct a confidence interval for whatever function of those parameters you're interested in. If you're a Bayesian, then the analogous procedure is to quote a posterior distribution of the function of model parameters that you're interested in, and I don't really want to get today into the sort of philosophical debate about whether you should or shouldn't be Bayesian. But the fundamental notion that we use probability as a metaphor for uncertainty and ignorance about truth, and therefore one needs to quote intervals which have interpretations in terms of probability or something rather like it. I've never quite bought the philosophical objection to confidence intervals because people think they're probabilities about the interval rather than parameter, but it seems to me operationally that's what you have to do. And the meta-analysis part of your question really brings us back, I think, to what assumptions are you making when you combine estimates. And so, again, there are models that people use for meta-analyses, which in their simplest form would assume that different trials are simply replicas of an identical experiment, and in their more sophisticated forms would allow both extant covariates attached to studies and latent random effects attached to studies to modify the precision of the final combined estimate. So, but the simple answer is a confidence interval or a Bayesian credible interval, according to your philosophical preference, is what I would want to see.
Okay. I've got another question here. So we're getting quite a few questions through. This is from Maria Costa. I'm going to direct this one towards Alan. It says, at the PSI conference, one of the sessions on estimands stressed the importance of separating the estimand from the analysis. However, Peter Diggle seems to think otherwise. I wonder what others think. So, as you are another, Alan, I'll give you the chance to first.
I think... So I think the framework that we came out, the estimand to some extent is separate from the analysis method. And the best way that we came out with that was we were defining estimands without really going into detail on the assumptions behind the missing data. So, but I think there's got to be a link between the objectives, the estimands, the study design, the analysis, and the sensitivity analyses. So I don't think you can do one totally without the other. But I tend to look at the estimand is trying to hone in on what is the real scientific question of interest here. That's how I'm viewing the estimands, not the technicality around is is missing at random appropriate as LOCF. It's about really what are we trying to do here? What is the basic question? That's how I'm tending to view estimands.
Yeah, could I just correct what may be a misconception? I absolutely agree. I mean, the way I put it is the most important thing that you do as a statistician is clarify with the person you're working with the real question. Now, that question may or may not be an estimation question, but if it's an estimation question, then estimand and the right question are synonyms. You've decided on what you really want to do. What you then want to do is actually, in my world, you want to build a model within which you can estimate the estimand and estimate its associated precision. But there is not, in general, in my experience, a uniquely correct model for a data set. And so in that sense, I'm not saying that you should model. What I'm absolutely not saying is that if you get a set of data, you first build a model for it, and then you say to the person who owns the data, what question do you want to answer? On the contrary, the first and most important step is to decide what question you answer. And you then need to select a model which does two things. It needs to include an answer to the scientist's question, and it needs to be not demonstrably incompatible with the data. But the question comes first and takes primacy. So I don't think Alan and I are in serious disagreement.
Okay. Moving to a question for me about design. So, clinical trials, you have the opportunity to design a trial, to choose the population that you're going to essentially sample from. And we talked about sort of longitudinal studies and dropout. What about other sectors? So are the estimand concept relevant to sectors other than drug development where you don't have design aspects, or maybe you don't have even longitudinal data, you're just looking to estimate things? So why this is happening more in drug development and not other areas, or are you seeing it in other areas?
So I've only I've spent most of my life in drug development, and so probably difficult for me to really make too strong a statement. But I think the principles, as Peter just said, that the principles of one of the key jobs of the statistician is help the teams to focus on what are the real scientific questions. But my limited experience going back 20 years in other areas, I think it still applies. I think some of the principles here still apply to other areas. Why is it taking off in the drug development? I don't know. The missing data discussion's clearly what's prompted it, but it's broader than missing data. Should it be in the other areas? Probably, but I don't have that experience. Peter?
Yeah, I think it's a universal. It's fundamental. It's basic. It's one of those things that's so basic that when I was first asked to take part in this meeting, I wondered why we were having a meeting on estimands. It's like saying, why don't we have a meeting on probability? I mean, it's just one of the fundamental ingredients of what we do. But it certainly isn't universal, as I've tried to indicate, perhaps not very successfully. Some problems that are often cast as estimation problems are really prediction problems. What I would guess as an outsider, that it's a necessary consequence of moving away from the absolute primacy of the test of the null. As soon as you start talking about estimating, you realize that life is both more interesting and more difficult. And so it's perhaps because of a change in culture within the pharmaceutical and the drug development world towards the increasing recognition that estimation is often more important and more interesting than testing that forces these discussions out into the open in a particular way, because you still have looking over your shoulder the regulatory bodies. And so that's always in the back of your mind, I guess. And now in science in general, I think exactly the same issues arise, but there isn't the same regulatory framework to social science or environmental science or basic biology. It's much more discovery science, and in that sense, there are fewer rules that you have to be aware of. So I think, in fact, the fact that you have the regulators looking over your shoulder in the drug development world forces you to actually be more honest, really, and be, less, therefore, susceptible to accusations of just going on fishing expeditions. than is true in some of the other sciences. So it's in a sense, you're being more rigorous in bringing the issue out into the open as an issue that you make it explicit rather than it simply being taken as read, that when you analyze data, you estimate things. What's the problem? And what you're doing, I think, very interestingly to me, is actually saying, no, let's really talk about this. Let's really bottom it out. And not just say, well, it's just a Greek letter or a function of Greek letters. What's the problem?
So if you look at developments at the moment in the big data area, say data science, where it's more about just collecting information that is being generated, in some cases passively, lots of it, and also maybe poor data quality. Certainly in the social sciences and survey areas, you can have 60% missing data. And then the advent now of machine learning coming to the fore. I mean, do you see this as being going in the opposite direction to that, or do you feel like really that we should see more of that estimand thinking coming into the data science arena?
I think very much the latter. I think the danger of machine learning approaches is that they tend, and I'm sure there are many honourable exceptions, but they tend to ignore context. And as soon as you ignore context, I think you're in danger of coming up with things which are technically brilliant, but dubiously relevant in the sense of being generalizable. I mean, I believe I've not been actively involved, but for example, there was a brief moment when everybody thought that Google flu trends was the answer to flu prediction, and then suddenly it went pear-shaped. And if there's no theoretical underpinning of what you do empirically, there's always a danger that it won't generalize. And I think, I think the data science and machine learning in particular have very impressive case studies in their ability to predict things empirically. But if you want to understand things, I think that's a very different world. And I would hope that we could come to some meeting whereby we can benefit from the really extraordinarily impressive technical achievements of the machine learning world, but also temper that with our more critical view of questioning at each stage, what are we really doing? What are the assumptions we're making? Are they valid? Can we actually have confidence that this finding is generalizable? So I would hope to see, and I hope maybe the RSS will have a role in this with our new data science section, which we hope will be launched in the near future, to actually find a common ground that intersects with those two cultures.
I've got a question here which sort of links into it a little bit from Kajira Rantel. And I think it pretty relates to using the term predictand, which I hadn't heard before. And we talked a bit about machine learning, predicting, etc. So the question is, can Peter clarify again the difference between estimation and prediction in the context of estimand?
Okay, the distinction I'm making here, and it's really just a way of using a word to make a particular distinction as to the things that a statistician is trying to do, which is that if the question that you're really trying to answer can be formalized as a statement about model parameters, then you're estimating. If the question you're really trying to answer involves a statement about what might happen if you were to observe something in nature that you have not yet observed, so it could be in the simplest case, for example, a forecast about tomorrow's weather, or it could be in a geographical context, a prediction of what would be the disease prevalence in a community that you've not sampled. You have data on nearby communities. In any context where what you're the question involves you understanding not only the model parameters, but also the random effects that are in the system that's being modeled. That's what I call prediction. So the difference is, are random effects involved in answering the question? If they are, it's a prediction question. If they're not, it's an estimation question. Okay. And that's why I personally am almost obsessive about using a different notation. Greek letters are parameters. Roman letters are random variables. They're random effects. And in a purely formal sense, the Bayesians don't make that distinction. Everything is just a random variable.
Okay, thank you. I've got one last comment here, which is from Jonathan Bartlett of AstraZeneca. It's a comment more than a question, or you might want to comment on it. Different estimands are definitely important and of interest. However, following Peter's comments a second ago, if there is no missing data, the ITT, or de facto analysis requires essentially no additional assumption. In contrast, all the other estimands try to predict what would have happened had the trial been different in some way, e.g. compliance had been different, receipt of rescue would have been different. It follows that analyses to estimate such estimands require additional, quite possibly untestable assumptions, unlike for the ITT estimand. So maybe Alan, I'll give you maybe the first chance of this. So any comments on that?
I'd like to read that one. My initial reaction is I'm not sure. I think the estimand is more than just about missing data. So going back to, you've got to link the question to things like non-compliance, treatment switching. So I'd like to read the question, but I'm not sure I agree with the statement as stated.
Okay. Peter?
Yeah, I think there's a distinction in my mind between missing data and treatment adherence. So as I understand it, ITT means that you in a simple parallel group trial with a single outcome measure, you take that outcome measure and you are basically comparing the means in a linear model under the assumption that the means are a function purely of which treatment you are randomized to. Right? Now, a corollary of that is that if there really are no differences between the treatments, you will get a valid randomization test, the null hypothesis of no difference, and a very good approximation to that is a t-test. So a very simple analysis will deliver the answer to that specific question. But it's certainly true that the extra requirements to estimate parameters in the presence of missing values, serial correlation, nonlinearity, and doubtless other complications does not come at zero cost. So it is certainly true that if robustness to assumptions is absolutely primary, and you have a simple cross-sectional trial with no missing values, I would just do a simple randomization-based analysis. Why take the risk? But as soon as you are in a more complicated situation, then it becomes more problematic and you need to have a discussion.
I think I've got one last question coming in here from ex-colleague of mine, Eddie Ekanaki, who's now at UCB in the US. Regarding use of estimands in regulatory submission context, a given trial may define various renditions of an estimand to address differing population needs. What considerations would drive the choice of what estimand to base a submission on, as in a regulatory submission on, and on what would one base assumptions of variability of chosen estimands for robust power? So I think this is more about choosing estimands based upon variability.
Yeah.
Or is the question about what label? So what are you targeting here? Doesn't the estimand go towards the label that you're targeting? And as I said, there's multiple stakeholders, so you're going to get multiple estimands in there. So I think you've got to look at that. So that's how I would interpret that one.
To me, relevance trumps power. You know, if the real question can only be answered approximately, that's better than a proxy question being answered precisely.
Agreed.
Variation on what you said famously.
Okay, well, I think with that, I think our time is up. So first of all, I'd like to say thank you to both speakers. So really appreciate that, and both the presentations and the QA. So thank you to Alan Phillips and Peter Diggle. Again, I'd like to thank our sponsors, Quintiles and Wiley. And I'd like to thank you all for joining today.